Okay, so I'm here to talk about a study of the clinical epidemiology and cellular genomics of diabetic retinopathy. So as mentioned, we're from the University of Cambridge. Welcome to our Sanger Institute, also collaborating with the London School of Hygiene and Tropical Medicine. So really this is a feasibility study. So we want to build cellular genomics models of human disease. So what we're aiming to do is collect rich and dynamic clinical information and biological samples so we can create a perpetual resource for cellular studies. So this would allow us to develop stem cells which we could use for derive into a range of different cell types, and we could do a range of cellular studies from comparing people with disease and without disease, or comparing what happens when you add a stimuli to those cells, or if you could test various drugs on this as well. So this is an overview of our recruitment strategy. So we're recruiting 3 groups of patients. Patients with diabetic retinopathy, patients with type 2 diabetes but without retinopathy, and general population controls. So what we've done at each GP practice, they run an algorithm at their site to identify on their clinical system potential participants. The GP then confirms the patient's eligibility, and then they mail out to the patients that are suitable to be invited. Patients then can call to the practice to book an appointment, and they have a single study appointment. At this appointment, they give informed consent. There's collection of blood samples so that we can extract DNA, and we can also later derive stem cells. There's also a limited web-based form data collection. We collect participant contact details, basic details on ethnicity, and their case control status. So we can later then integrate this DNA stem cell derivation along with the recorded data. And once recruitment is complete at the site, we will then extract the electronic health record data on relevant records for the patients, so on their diabetes or the diabetic retinopathy. So our experience of recruiting in primary care has actually been really positive. We've had a lot of support from particularly the Eastern CRN, and they're actually, the research nurses there are actually conducting our study appointments. We've actually had good engagement with the practices and the site study teams as well, and we've had a lot of enthusiasm to participate from CRNs over the country, and also GP practices when we've been to GP forums set up by the CRN. There's been a lot of enthusiasm to participate in the study. It's also been fairly straightforward once we had the sort of overall approvals through like ethics, HRA. Because we've had approvals at the regional level in the Eastern region, we've been able to fairly quickly bring on board the new practices. The one thing that we're working on at the moment is the electronic health record transfer. So by what method can the practices send us those records so that they're sent securely? Their preference would be by NHS email, but we're not part of the NHS directly, so we don't have that method. So it's setting up another method where we could securely transfer those records. And this is really just, my last slide is a bit about where our group is sort of visualising our research going forward. So really one of our things is to build a large resource, maybe of up to 1 million people, where you could integrate genomics data, clinical data from electronic health records, and you could do discovery and innovation research. The second thing is looking at integrating genomic records into clinical care, but also thinking about evaluating that. How well is this information used? How well does this work? So quite a lot of evaluation around that. And thirdly, really engaging patients with their information. So allowing them access to some of their clinical records, some of their data, so that they can be engaged in an informative and accessible way to this kind of data. That's it. Thank you very much.